Yes, good evening. Where we have stopped in the last class. Incomplete dominance. Yes. Monohybrid cross and incomplete dominance we have done. Yes, today we will study two gene inheritance or dihybrid cross. Yes. So Mendel, when he conducted the experiment for one gene, now he proposed two laws, that is law of dominance and law of purity of gametes or segregation of gametes. So when we recall the law of dominance, the law of dominance says when two genes are present together, one gene will be dominant in nature and one will be recessive in nature. The dominant gene will suppress the recessive gene, because of which the dominant gene will express itself and recessive gene, in spite of present, it is suppressed by the dominant gene. Law number 2, purity of gametes or segregation of gametes. According to this law, Mendel says when a pair of genes or a pair of allele, when they are present together in the F1 generation, if blending is not taking place, when the blending is not taking place in the F2 generation, when the segregation takes place, the gamete segregates, and that proves the purity of gametes. So two laws were proposed by Mendel. After this, one more experiment, very important experiment conducted by Mendel, that is two gene inheritance or dihybrid cross. So here the word itself is telling you Mendel has taken two genes. Okay? He has taken two genes for conducting the experiment. One is shape and one is color. Two characters he took in common. One is shape, one is color. When he took shape, what are the two possibilities for shape? Yes, one we will take it as round and one we will take it as wrinkled. One round, one wrinkled. And two colors, one yellow and one green. Yes. So in this, one will be dominant, one will be recessive. So to conduct the experiment, Mendel took the parental generation. He took the parental generation. He took two plants. One is homozygous round yellow and one more homozygous wrinkled green plant. Two plants he has taken with one shape round, one shape wrinkled, one color yellow, one color green. If you take the phenotype of this, the phenotypes will be round and yellow. And here the phenotype is wrinkled and green. So here round one shape, wrinkled one shape, yellow one color, green one color. If you take the genotype of this, take the genotype of this. For round, we can give the symbol as capital R, capital R. And for yellow, we can give the symbol as capital Y, capital Y. For wrinkled, we will give the symbol as small r, small r. And for green, we will give the symbol as small y, small y. Yes, this is the genotype of shape and color. Then take the gametes from it. So one gamete we will take from here, from the capital R. One gamete we will take from capital Y, capital Y. One gamete with small r, and one gamete from small y, small y. Yes. Then we need to go with hybridization process. Yes. So when you cross this all the four together, the process is called cross pollination. So what is the result we get? Capital R, small r, capital Y, small y. Yes, this is the F1 generation. This is the hybrid we have got by crossing four plants with two characters, shape, color. One is round, one is yellow, one is wrinkled, one is green. So here, what is the result we get, sir? We will get heterozygous round yellow. Yes. Did you understand till here? Yes. So here, till here, law of segregation of gametes come into existence. According to the law of segregation of gametes, when a pair of genes, when a pair of alleles are present together, all will express itself in the F1 generation. We are able to see they are expressing together. No? They are expressing. And here, what segregation tells? When they segregate, when they separate, no? All the gametes will separate because blending does not take place, mix up does not happens, because of which segregation of gametes take place. So gametes are pure. This was proved. Now, with this F1 generation, if we go with the F2 generation, Shall we proceed? Shall I erase? Yes. What we have to do first? Capital R, small r, capital Y, small y into capital R, small r, capital Y, small y. Yes. Now what we are doing is self pollination. Earlier what we have done is cross pollination. Now we are doing self pollination. Eh? So here if you take the possibilities, see, if you take the possibilities, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Yes. So from one plant, four possibilities come into existence. See, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So these are the four possibilities we got from one hybrid. Okay? We have done the hybridization by self pollination with one more plant. Right? So again we will get capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Four possibilities here. Yes. Now, if you cross capital R, capital R, capital Y, capital Y, R, R, Y, Y. Done? Yes. What is this capital R, capital R, capital Y indicates? Yes. Round, round, yellow. And this is in homozygous condition. Yes. Then capital R, capital R, capital Y, small y. This is also round, yellow, but in the heterozygous condition. Then capital R, small r, capital Y, capital Y. Again this is round, yellow in heterozygous condition. Then capital R, small r, capital Y, small y. Round, yellow in heterozygous condition. Did you understand till here? Yes. Second, coming to the second row, second column. Capital R, capital R, capital Y, small y. Capital R, capital R, small y, small y. Capital R, small r, capital Y, small y. Capital R, small r, small y, small y. Yes. Now capital R, capital R, capital Y, small y. Again round, yellow in heterozygous condition. Then capital R, small y. Yes. It is homozygous or heterozygous? What is capital R, capital R? Round. What is small y? Green. Homozygous condition, heterozygous condition. Yes. Homo or hetero? It is, how it is homo? Huh. If it is RR in capital, it is, if both are small y, then it is homo. Since they are different, it is heterozygous condition. Then round, yellow, heterozygous condition. This is also round, yellow and heterozygous condition. Did you understand till here? Did you understand? Yes. Next. Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Small r, small r, capital Y, capital Y. Then small r, capital Y, small y. Yes. What is this capital R, small r, capital Y, capital Y? Yes. Round, yellow, heterozygous. It should come first, but I have written down to just make it convenient. Yes. Again, this is round, yellow in heterozygous form. Then this is, yes, wrinkled yellow. Which form? Heterozygous form. This is wrinkled yellow in the heterozygous form. Then capital R, small r, capital Y, small y. Then capital R, small y, small y. Then small r, capital Y, small y. Small r, small y, small y. Yes. Again, this is round, yellow, heterozygous. This is round, green. This is also round green. Heterozygous and wrinkled yellow heterozygous form. And this is wrinkled green homozygous homozygous wrinkled green. Yes. So here, by writing this, filling this Punnett square, this is called as Punnett square. Okay. A scientist by name Reginald Punnett. Reginald Punnett was the person to make this square, which is called as Punnett square, or also called as checkerboard. Okay? Yes. So with this, we study one more law, that is law of independent assortment. What is the law of independent assortment? According to the law of segregation, in the F1 generation, if blending does not take place, if blending does not take place, the gametes will segregate apart. They are pure, they have not mixed up. So this is a continuation of law of segregation. That gametes which segregates apart will occupy the particular position in the Punnett square. They will occupy the particular position. See, we have taken capital R, small capital Y, capital Y. This was our parental plant. We have taken small r, small y. Okay? We have taken this parental parent. They have occupied two positions. Ne? Did we take round green? Did we take round green? No. Did we take wrinkled yellow? No. Then from where it came? It means during the segregation of the gametes which were present together in the purity of gametes, now they have got the opportunity to segregate themselves and occupy particular position. So when they occupy particular position, that is independent assortment. Is that clear? After segregation, the gametes have independently assort to the particular position given to them. So therefore, we will get the result like this. If you tally, no, we have round yellow, wrinkled, yes, wrinkled green, round green, wrinkled yellow. 4 we have got. Right? In this 4, the parental generation was round yellow and wrinkled green. These two which have been segregated, have expressed themselves by assorting a particular position in the Punnett square. Okay. Now, if we take the phenotypic ratio round yellow is to round green is to wrinkled yellow is to wrinkled green. 4. No? So how many round yellow we have? See, 1, 2, 3, 4, 5, 6, 7, 8, 9. Totally 9 we have. Then round green we have 1, 2, 3. 3. Wrinkled yellow, how many we have? 1, 2, 3. 3. Wrinkled green, how many we have? Only 1. Ne? What is the ratio we got? 9 is to 3 is to 3 is to 1. This is the phenotypic ratio. Okay. This is the phenotypic ratio. If I ask you, write the genotypic ratio for this, will you be able to write? Yes? Not possible? Do it, we'll see. What will be the ratio? Write down fast. Hello. Then what is the ratio? Now tell me the ratio. Genotypic ratio you tell. What is the ratio we get? What is the ratio? Hmm? नहीं करे? हाँ? वहाँ कुछ स्टॉप कर डाले? कॉलेज में नहीं करा है? हाँ? नहीं करा है. हाँ. In the seat, you need usually what happens? You need to write the genotypic ratio also. नहीं, कब तो भी genotypic ratio पूछ डालते हैं. That time how to calculate? Easy method. Two things. Two things. One, by taking the genes, gametes. Okay. And one, in short, we can tally. How is that, sir? See, how many round yellow are there in the first column? 1, 2, 3, 4. Yes, 4 is 2. In the second row, how many are there? 2 are there. Yes. In the third row, 2. In the last row, 1. Yes. Then round green, it is present in the first row? No. In the second row, how many are there? 2 are there. In the third row, it is present? No. In the fourth row, how many? 1. No? Wrinkled yellow, first row it is present, second row it is present, third row, how many? 2 are present. In the last row, 1 is present? Yes. Wrinkled green, 1 in the last row it is present. Now, this is the ratio 4 is to 2 is to 2 is to 1. 2 is to 1, 2 is to 1. See? 9, 3, 3, 1. It is correct? Yes. This is the genotypic ratio. Done? Did you understand? Any doubts you have till here? Yes. Shall we go with the test class now? For this? Yes. For the test cross, what we have to do? We have to take the F1 generation, capital R, capital Y, small y, into recessive parent, small r, small r, small y, small y. Yes. Done? Now, if you cross this, how to cross? We have to take one gamete. Nay, take the possibility capital R, capital Y. Nay, capital R, capital Y. Then capital R, capital R, small y. Small r, capital Y. Small r, small y. Done? Yes. Now, if you take the possibility for this, we have to take one time or take two time. Both are same. Nahin? So here if you take small r, small y. So here capital R, small r, capital Y, small y. Then capital R, small y, small y. Then small r, capital Y, small y. Then small r, small y, small y. Yes. If you see what is this capital R, small r, capital Y, small y, it is heterozygous round yellow. Done. Yes. Now this is heterozygous round green. Done. Yes. This is heterozygous wrinkled yellow. And this is homozygous wrinkled green. We have taken one parent, we got back one parent. Yes. Yes. What is the ratio? 1 is to 1 is to 1 is to 1. Correct? Yes. Test cross is proved. Whatever the experiment we have conducted, that is correct. 1 is to 1 is to 1 is to 1 is the ratio. Today topic completed Done? Any doubts you have? So how many we have finished? 3 we have finished. One gene inheritance, incomplete dominance, two gene inheritance. Done? Shall we proceed with the fourth one? Yes. Fourth very important topic we have co-dominance. Co-dominance. Yes. What is this co-dominance? Any idea? Yes. Co-dominance. No. We have studied incomplete dominance. What did we study in incomplete dominance? What did we study? two genes are present together, both are unable to express themselves. Neither of the gene are dominant. Because both are not dominant, hybrid, that is intermediate hybrid, was produced as the F1 generation. Yes, we took the example of snapdragon flower, okay, 4 o'clock plant as the example for incomplete dominance. Now, in the codominance, both the genes, both the genes or alleles try to express themselves together. Both the genes are trying to express themselves together. Very good example we can take is ABO, blood grouping. Blood grouping in man. ABO, blood grouping in man. Now, first thing what we have to understand during the codominance is, in the codominance we are able to see expression of more number of genes together. Both the genes are trying to express itself. So here, when more than one gene try to express itself, it is called as multiple allele. Multiple gene or multiple alleles trying to express themselves. करे भाव So here, when the genes or alleles more than one, if they are expressing themselves, that means the expression of trait is controlled by many genes. Here, A is also trying to control, B is also trying to control, O is also trying to control. So this process or this phenomena is called multiple allelism. It is called multiple allelism. Done? Yes. So in this ABO blood grouping, what we have to understand is, our blood is controlled by I gene. Our blood is controlled by I gene. Okay. A gene by name isoagglutinogen gene. A gene, I gene means isoagglutinogen gene. So this I gene, isoagglutinogen gene. So here, if you see the RBC cell, RBC cell, the surface membrane of the RBC cell consists of antigen. It consists of antigen, and the plasma which is present, the plasma consists of antibodies. Plasma consists of antibodies. Antigen and antibody will not be same. It will not be same. Now, assume the blood group. The blood group is A. Now, the antigen will be antigen will be A and antibody will be B. Okay. If it is B, it will be A. Correct? Yes. If it is AB, antigen will be AB. Antibody will be nil. Okay? And if it is O, the antigens will be nil and antibody will be O. Done? Yes. See here. Now, what to understand here? This I gene, which will control our blood, our RBC, red blood cells. So, this I gene has three allelic form. Okay, three allelic form. It is showing polygenic inheritance. It is showing the mechanism of polygenic inheritance. Means many genes are expressing their expressions. Many allelic forms will be expressed. See, one gene will express IA, IB and I. One isoagglutinogen gene is expressing three allelic forms: IA, IB and small I. Here, see, this IA and IB are having sugar. Here, sugar is absent. If IA is present along with IA is dominant. If IB is present along with IB is dominant. Nay? Because this is recessive. Nay? This is, did you understand till here? Now, take the condition. See, two patients we will take. First person. Second person. Yes, first person and second person. So here, if the blood group is A, blood group is A, this person will have I A. Okay. If the second person is having I, nay, what will be the genotype of this? Huh? I A I. Done? Yes. If it is blood group B, nay, I B I. What will be the genotype? Yes. Did you understand? If it is O, nay. So here it is I, correct? Huh? For O, what it should be? Correct? Yes. Done? Yes. Now, if it is AB, no? I, A, I, B, I, A, I, B. Now, what will be the result? I, A, B, I, A, B. Here, this is dominant, this is recessive. This is dominant, this is recessive. Both are recessive form. Okay, here both are dominant. Nay, both are able, A as well as B are able to express themselves at the same time. Yes. So this AB, no, is dominant, showing co-dominance. They are able to show co-dominance. Nay, a pair of genes are present, a pair of alleles are present. Nay, when one is expressing itself, one is getting suppressed, dominant and recessive, dominant and recessive. At a time, one is able to express. Nay, if both are able to express, it is codominance. Okay, both are able to express at the same time. The phenomena is called as codominance. Did you understand? Yes. This is the usually this will be asked for 2 marks. What is the genotype of A blood group? We have to write IA, 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 I. ठीक है? This is the genotype of A blood group. Two parents if we take. ठीक है? If it is IB, 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 I. ठीक है? If it is O, I. If it is AB, I, AB, I, AB. Done? Did you understand? Yes. Hmm. We'll finish and go. Huh? Okay, na? Hmm. Yes? No, no. Why it is not 3 IA like not like that? Okay. See, when in 1900, a person by name Karl Landsteiner, he was the person to discover our blood grouping. Okay. Blood grouping was discovered by Karl Landsteiner. When he studied the blood grouping, no, he studied the red blood cell first, RBC cell. Okay. He was able to see that RBC cell has two types of proteins. Okay. One is called as antibody and one is called as antigen. Both are proteins only. Okay. But usually antibody it's a positive protein and antigen no sometimes it's a positive protein sometimes it becomes a negative one. It can be an enzyme also. Okay. So he studied that the surface membrane of the red blood cell outermost covering consists of antigen and inside the plasma contain antibodies, and he was able to study antigen and antibody in the blood group will not be same. It will not be same. Then he studied that blood grouping, why it is A sir, why it is B sir, why it is O, why it is particularly AB. No? So this blood grouping which we come, it will come to us. Nay, it is coming through our parents. If your blood group, assume you are O positive, that will be from your mother or from father or from both. Okay. It's not like you will be A, your mother will be B, and your father is AB. No. It is from the one parent you will get. So that blood group that comes from the parent to the offspring right, is controlled by three genes. It is having three genes. Okay. That gene is called as I gene. In the blood group, we have one gene that is called as I gene. And that I gene is showing polygenic inheritance. What is polygenic inheritance? Means multiple allelism it is showing. Okay. Expression of the allelic form is by many genes. Okay. So there we are able to see that I gene with A, I gene with B, and small I. That is IA and IB are having sugar. Okay. On the red blood cell we have some amount of sugar, and I small I does not have. That small I indicates O. Okay. So here because only antibodies are present in the O blood group, antigens are not present, we will call it as universal donor. Okay. O negative is universal donor. O negative blood, yes, they can also give, but only O group with only antibodies and no RH group also. RH factor is also not present. Plus or minus RH factor that is also not present. That means they can give to anyone. They are called as universal. Now AB positive, AB positive. They are called as universal recipient. Why they are called as universal recipient? They can take the blood from A also, B also, AB also, O also. Okay. Therefore, they are called recipient. Now here three genes are present. If the combination, if it is IA with I, A is dominant, small a is not dominant. If it is B, B is dominant. Nay. If I take two parents, I have taken here, I have taken only one gene, two genes I have taken. Here I have taken alleles. This is one allele, this is one allele. Okay. Individual if you take, nay, if I take this is a gene. If I take this together, it becomes allele. Nay, I have taken the genotypic ratio. Nay, so that genotypic form we have to take for allele. So I have taken this is one. This is one. Assume instead of this, see, it is IA, IA. Nay, what will be the result here? It is again IA, IA. If it is IB, IB, it will be IB, IB. Nay, we are able to see only dominance. When it is codominant, when both the genes are able to express or both the alleles are able to express at a time, okay, then it is called as codominance. Good example is ABO blood grouping in man. Is that clear? Did you understand? Yes, two very important topics we have studied. One is dihybrid cross, and second is codominant. Done? Hmm. Oh? Yes, he can give, but he cannot. O negative. O negative, AB negative. Okay, it's a rare match. AB negative and O negative are related. Negative usually because if the RH factor is not present, then it is like that. Okay. RH factor will be present. Why? Because either in the mother or in the father, that factor will be present and that will be transferred. Therefore, majority will have A positive, or B positive, AB positive, O positive, and positiveness comes from one side. In case if both the parents are negative, then only it will be an issue. Okay? Did you understand? Is that clear? Thank you. Any other words you have? Oh, no, theory of inheritance. Then we'll go with the determination. Then we'll go with the disease. Genetic disease. Huh?